Hello everyone. Today we will look at the structures and functions of the seminiferous tubule in the testes. Let's begin with a clinical case. John, 29 years old, and 28-year-old Rose have been married for 3 years. They wanted a baby, but Rose could not get pregnant. After examination, doctor told them there were anti-sperm antibodies in John's blood, which could be the reason for the infertility. The questions are, what is normal process and circumstance for the development of sperm? How does the body normally prevent the production of anti-sperm antibodies? At the end of this session, you should be able to answer above questions. List the general structures and functions of the testes. Describe the circumstance for the development of sperm, and explain some associated clinical changes. In the testes, besides the capsule and the septum constituted by connective tissue, the main structure of testes is convoluted seminiferous tubules, short string tubulus, and dilated rete testes. The main functions of the testes are to produce male gamete, sperms, and male hormone, androgen. The seminiferous tubule in the testicular lobules is the key place for these two functions. Shown here are the seminiferous tubules stained by hematoxin and eosin. Androgen is produced by the interstitial Leydig cells found between the seminiferous tubules, while sperms are developed within the seminiferous tubules. The lining cells of the seminiferous tubules comprise the complex stratified seminiferous epithelium, with two basic cell populations, spermatogenic cells and Sertoli cells. Before puberty, the spermatogenic cells are all at the stage of diploid spermatogonium. After puberty, the spermatogonium began to undergo differentiation. From the basal membrane to lumen, we can find the different development stages of the sperm: spermatogonium, primary spermatocyte, secondary spermatocyte, spermate, and the mature sperm. The Sertoli cells are not part of the spermatogenic cell lines, but they provide support for the nurture the development. They are easily identifiable by their nuclei in hematoxin and eosin stain slides. The nucleus is pale and oval to pyramidal in shape, with one or two prominent nucleoli. The Sertoli cells have a very extensive and branched cytoplasmic structure. Little of its outline can be seen under light microscope. However, the extensive branching nature of its cytoplasm can be identified under the electron microscope. This diagram shows us the ultra-microstructure of the Sertoli cells and their relationship to the wall of the tubule and to other cells in the seminiferous epithelium. In this picture, we can easily identify the cytoplasmic outline and the tight connection between them. The connection between the adjacent Sertoli cells dividing the wall of the tubules into two parts, luminal compartment and the basal compartment. The basal compartment near the peripheral region is sealed off from the luminal compartment by the processes of adjacent Sertoli cells. The diploid spermatogonia are found in the basal compartment. After the spermatogonia start division and differentiation, the Sertoli cells send out new processes to undermine the forming primary spermatocytes and seal off a new layer below them, then break the barrier above. This effectively transfers the differentiated haploid cells to the luminal compartment. Why is this elaborate mechanism for separating the tubule into two compartments necessary? The basal compartment has access to the bloodstream and all the immune components it carries. The spermatogonium and the Sertoli cell have been there from embryonic stage, so the immune cells are familiar with them. When a boy reaches puberty, the diploid spermatogonium differentiates into haploid primary spermatocyte, secondary spermatocyte, and so on. The differentiated haploid cells express larval proteins on their surfaces. There is a risk that the immune system might recognize them as non-self and produce antibodies against them. So, for self-protection, the blood-testis barrier is created by the junctions of adjacent Sertoli cells. Maintenance of two separated compartments allows the different stage of the spermatogenic cells to exist under optimal conditions. The blood-testis barrier consists of the tight junction between the Sertoli cells, base membrane of tubules, interstitial connective tissue, endothelium, and the capillary basement membrane. Among the structures of blood-testis barrier, the tight junction between Sertoli cells plays the most important role. However, the blood-testis barrier can be disrupted by physical or chemical injury or by infection. When this barrier is broken, differentiated sperm antigens escape from their immunologically protected environment and come into contact with blood elements. That may initiate the lymphocyte to produce anti-sperm antibodies, which immunologically attack the differentiated haploid cells in the seminiferous tubules. Anti-sperm antibodies can be present in many men without necessarily inducing infertility. However, some of them may have reproductive problems. For example, June in our case. Besides being involved in the formation of blood-testis barrier, Sertoli cells also have other functions. Firstly, the cytoplasm of Sertoli cells can form a network to provide general support, protection, and nutritional supply for sperm development. Secondly, they have the function of phagocytosis. During spermatogenesis, excess spermatid cytoplasm is shed off as residual bodies. These cytoplasmic fragments are phagocytosed and cleared by Sertoli cells. The third function of Sertoli cells is to secrete inhibin, activin, and ABP. Androgen-binding protein ABP can serve to concentrate androgen in the seminiferous tubules, which is necessary for the development of sperms. Let's summarize the structures and functions of the seminiferous tubules in the testes. Seminiferous tubules are highly convoluted structures within the testicular lobules. The lining epithelium is mainly composed of spermatogenic cells at different development stages: spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, and sperm. The Sertoli cells are also within the lining epithelium of seminiferous tubule. and play important functions in spermatogenesis. These are the references for this session. Thank you for your attention.